So then, without further ado, then Xavier Altafaj will start today. Xavier is located in Barcelona, has been involved from very early on with the association, with the fundamental basic research into some of the early identified mutations, and has helped us a great deal to move everything ahead and explaining the background of the GRIN mutation, its clinical manifestations, etc., to all the parents involved. He's a professor at the University of Barcelona and is a part of the Barcelona GRIN team group as well. So thank you very much, Xavi. I know you're going to give a beautiful overview today, and we will get started with you. I just want to also introduce Mireia Olivella, because we're going to go smoothly from one speaker to the next. Mireia, we know from our own independent research work in other fields, and I had the luck to get her involved into GRIN. And she is a professor in bioinformatics, the best structural chemist I've ever met. And she is really the brain behind those structures that you're going to see that tell us so much about the potential impact of mutations on the function of the NMDA receptors. Professor at the University of Vic in Central Catalonia, and also a team member of the Barcelona GRIN group. So thank you, both of you, and please, Xavi, I'll pass it over to you then.
Okay, so thank you very much, Christian, for this kind introduction of Mireia and myself. So I'm going to proceed to share my screen. And if everything is fine, and you can see in the whole screen the presentation, I'm going to move forward. Okay, so thank you very much, especially the organizing committee for preparing this wonderful scientific program for today's session. Thank you especially Sandra, Mel, and Marita, but also all the people from the Green2B Europe for organizing this meeting, this virtual or remote meeting that at least can provide you the new results and data that have been obtained by several groups along the last year. I think that that GRIN community is growing, not only in terms of families, but also the associations are growing up, and also research and research networks are being formed. So this is the most promising aspect, I mean, from the last year, that people and researchers and families are pushing up research and translational studies. So for today, I try to give you an overview on what is NMDA receptor, but at the same time, I try to avoid being too redundant with previous presentations. For those of you that already attended my talks in the first and second days of the conference, I completely changed, well, not fully, but almost, the presentation. And I wanted to give a view probably more integrative, since I think that the field of NMDA receptors and GRIN-related disorders is really focusing on brain phenotypes. But I think that, and you, the families, you are very aware of that GRIN-related disorders goes beyond what are the neuronal phenotypes. And please let me go to the origins, or not the origins, but at least ancient times. This is a draw from Leonardo da Vinci. This is the Vitruvian man. And I wanted to start the talk by presenting this image, this illustration that somehow shows that the proportions in a body are really they can fit this circle, meaning that this is something that is like the geometrical shape of the perfection. And also this really, at least for me, it gives me this view that all the systems in a human body are closely connected. So I think this is very, very important, not only in health, but also in GRIN-related disorders. So we need to go beyond what is just the brain phenotypes and try to connect what are the GI tract phenotypes, what are the motor alterations that can result from NMDA-receptors dysfunctions. So from this very initial cartoon and view of what is a man or a woman, of course, along the centuries, the technological developments have provided very valuable tools to understand and to go in very much detail on what is really happening and what is health and what are the disease conditions. So this zooming in has been achieved by many technological developments. In the right part of the slide, you can see a human. So this is a length dimension. Okay, so you see that a man is it's in between 1 to 2 meters height. And if we go more in detail and we use microscopy, we can have different images or different, we can have different levels of study, what is really inside man. So we have the systems, liver, brain, heart, but then we have the basic unit of human beings that are the cells, okay? And even more in detail, cells are composed by several, by a complex machinery that is represented here, such as mitochondria, okay, and there is endoplasmic reticulum, plasma membrane, the nucleus, and if we go even more in detail, there is DNA. So currently, we have a lot of information on DNA, and this is what we obtain, or families have when they have the genetic report. But then, what is difficult is to understand how DNA will determine what is happening in a human being. And in the context of GRIN-related disorders, how DNA variants can trigger some neurological conditions and systemic conditions, as in GRIN-related disorders. So brain is a very complex system that somehow is using electricity to connect millions of neurons, millions of cells among them. And this structure is really powerful and is controlling most of or at least almost all the systems of a human body. So brain is not only involved in what we call the neuronal phenotypes that can be represented by these emojis. So the emotions, communications, and cognitions, of course, they are closely related with brain function, but also senses. So senses are allowing us to communicate and to have this adaptation to environmental stimuli. So senses are necessary, well, to have vision, to hear properly, for smelling, taste, and touch. And brain is integrating these senses, sensorial information, and to providing an adapted response. But also brain is important in the sleep pattern and also controls the immune system. So whenever there is a brain alteration, since brain is like the master commander of the human body, then sleep patterns can be altered. This is something that will be visited by Dr. Garcia Cazorla, and sleep pattern is also one of the phenotypes that are affected in GRIN-related disorders. Further, immune system is also modulated. There is a cross talk between immune system and brain. And brain also is important for the control of movements and the gastrointestinal function. So there is a very close relationship between gut and brain, and this is a bidirectional connection. So from this initial and central picture of the brain, what you can see here in the upper left part of the slide is this connectivity, these pathways. These are like highways that connect brain with the different organs of the body. This complex circuitry was already described, and this is a real illustration by Santiago Ramón y Cajal, a Spanish neuroscientist, that defined, that drew this neuronal network and this connectivity that you can see here. So he called these structures the butterflies of the soul because of their butterfly shape and their beauty as well. And one important thing that you can see here is that these neurons, you can see this central part, which is corresponds to the soma, including the nucleus and the DNA. But all these thin processes here, which are called dendrites, are indeed creating a very complex network. So they are like a spider net that are connecting thousands of neurons amongst them. And the context, the very thin context, indeed correspond to this particular structure, which is called the synapse. So a synapse is a part of the neuron, or of two neurons that are very close one to each other. And this allows a crosstalk. So when one termination of neuron 1, such as here, has a stimulation, it will release what is called a neurotransmitter. A neurotransmitter is a chemical compound that is released. So it's like sending a message from neuron 1, and then neuron 2, that is very close, can sense this neurotransmitter. And upon this release of the neurotransmitter, the receptors that are in neuron 2 will catch the neurotransmitter, and they will somehow understand that they must be activated. This is important in the context of NMDA receptors, of course. So here you can observe the neuron 1 that is releasing the neurotransmitter at this level, the synapse, and then is passing the information, okay? So this is a flux of information between synapses. So specifically, and now we are focusing on NMDA receptors. NMDA receptors, as you know, they are receptors for a neurotransmitter, which is glutamate. And NMDA receptors are very complex proteins that are able to sense glutamate. They are composed by different, what we call subunits, different proteins. Some of them are GLU-N1, GLU-N2B, and these proteins result from are the gene products of GRIN genes. So in other words, in our cells, we have DNA. This DNA has thousands of genes, and some of these genes are GRIN genes. GRIN genes, they are transformed into proteins, and these proteins build up NMDA receptors. But what are really doing NMDA receptors in these synapses, in these neurons? So here you can see that when glutamate is released in the synapse, this allows an opening of these NMDA receptors. Somehow this is like a door that is being opened. And when this door is opened, this allows calcium influx. Calcium goes into the neuron too. And this calcium is really very, very, very important for neuronal physiology, okay? So this calcium is like a green signal, a green light that may allow the neuron to respond. Okay, so, but this is what we call under physiological or standard or normal conditions. But what is happening when there is a variant, a mutation in these GRIN genes? The different mutations are roughly classified into two main types. We call the first type the gain of function, okay? Gain of function meaning that they have an additional function. But in that case, a gain is not something, it's not a quality, okay? It's not an asset. It means that this gain of function indeed corresponds to an additional function, and then this can have a negative effect on NMDA receptors. So let's see in this cartoon what is happening when there is a gain of function variant. Please remember that when there is, in the normal conditions, okay, there is this influx of calcium. This influx of calcium is regulated. Here we represent it by two calcium ions, two green balls that are going inside. But in gain of function variants, in addition to this normal calcium influx, there is an additional calcium influx. This is in red, okay? There is too much calcium that is going inside. And too much calcium means that something will go wrong. There is a second type, a second classification of GRIN variants. And these are called the loss of function, meaning that they have less activation, okay? So in that case, we have, in comparison to the normal NMDA receptor, loss of function GRIN variants, they lead to a reduced calcium influx. There is not enough calcium going through these receptors when they are activated. So with an analogy to a river, this in the left part, you will see the receptor that is opening a door, or yes, and then is allowing this influx of calcium, or in that case of the river, a normal rate or flow rate of water. In gain of function, there is too much water that is flowing, and this can be can be negative. In the contrary, for these GRIN variants that result in loss of function, there is not enough water flow. So in both conditions, it is really important, and when there is a GRIN variant, it's very important to determine whether this GRIN variant, a specific GRIN variant, will result in either a gain of function or a loss of function. This is the initial and what we call the functional annotation of GRIN variants. And is really important because in terms of therapies, what we and others will try is to normalize, okay, to give these receptors that are either gain of function or loss of function to reestablish normal flow rate of calcium. So the first and most critical part is to determine after the genetic diagnosis whether a specific GRIN variant leads to a gain or to a loss of function. So this is a summary table from this initial part of the talk. We have in normal conditions here in green, we have a green gene that results in a gluon protein and NMDA receptors that is allows this calcium influx, and this is important for neurotransmission and brain function and all the systems control. Okay, this is under what we call physiological conditions. In the presence of a green genetic variant that is pathogenic and can result in a loss of function or a gain of function, there is an alteration of gluon protein that results in NMDA receptor dysfunction, and neurotransmission and brain functions are altered. So the difficulty of GRIN-related disorders is that from a few GRIN genes, there is multiple elements, multiple DNA elements that can be affected, okay? And this is something that Mireia will illustrate in the next talk. And at the end, this results in a very diverse clinical spectrum that Ángeles García Cazorla will show you in the third talk. So our pipeline, and this is also something that is being used and followed by most of the groups working on GRIN-related disorders, is that from a genetic diagnosis, families or clinicians are contacting people that is performing these functional annotations. In the Barcelona GRIN cluster, we perform these annotations using in silico or computational studies. These are led by Mireia Olivella, and also in vitro studies. We record the currents and we study what is happening when these variants are expressed in cellular models. This allows to classify these GRIN variants into gain or loss of function variants. And this is really important for decision making of precision medicine. So we are especially interested to evaluate new therapeutic strategies that can rescue either gain of function variants by attenuating them, their activity, and loss of function variants through the enhancement of NMDA receptors. So overall, this is like an inverted pyramid in which from many GRIN variants at the end, we want to focus and to define which treatment will be the most optimal for each individual case. So once the families or clinicians have the genetic report, in the genetic report, basically it will give a milestone on which part of the DNA and which nucleotide is effective. This information is somehow difficult to interpret by families, but this is the most important information if we wanted to start the functional annotation. So from this in genetic report, you can contact our team or others and request a functional analysis. So the second, once you contact the functional analysis laboratories, it is important to provide the genetic report, also to establish physicians' contact, okay, because they will be also the relay between you and researchers and physicians. Also very important to provide an informed consent signature. And then with all these documents, the laboratories can start the functional annotations. And after, depending if this variant is annotated or not, the time to deliver the functional report will vary. So this functional report will, at the end, be sent to the physician that is following GRIN children, and clinical decisions will be adopted based on the functional reports. So we currently have about more than 150 cases and families and clinicians that contacted us, and we started to perform most of these functional annotations. The distribution, at least this is for Barcelona GRIN cluster, okay, and most of them are from Europe. About more than 100 are from Europe, but recently we have also been contacted by several clinicians from South America and also from the States, Canada, and many other countries. So once we have this genetic report that is provided by families or by clinicians, then we start a first set of computational studies, which will be presented by Mireia. And also, if the GRIN variant is not already reported, we undergo the functional annotations of GRIN variants. So this is the work that we are doing in our laboratory. I would like to mention the work of Ana Santos, who is here and who's doing an amazing work in cloning and expression and functional annotation of these variants. So she's a brilliant PhD in my lab, and she's a really hard worker, very organized, and she has already annotated more than 50 different GRIN variants. But also, I wanted to acknowledge especially Fede Miguez, a PhD student, and David Soto, who are also the masters of electrophysiology. So this is a very time-consuming task. Okay, so we need a few days or even few weeks for each individual variant to be properly annotated. And what we do normally is to express, so to create the GRIN variant of interest, to express in cellular models, to see if this variant goes to the membrane or is expressed in the synapses, and then we move to the electrophysiology. So basically, we measure how these receptors, how these NMDA receptors doors, let's say, how they can be activated, and if they are a gain or a loss of function. So recently, I would like to, I'm happy to show you this slide, the next two slides, in which a part of these annotations have been recently the object of a manuscript that has been recently accepted for publication. And this one consisted in the annotation of a particular set of GRIN variants that indeed is what we call the truncations, okay? So these GRIN variants, as you can see here at the bottom, the green ones here, okay? These truncations result in shorter NMDA receptor subunits, okay? So it's like the protein is cut and it's not integral. And what we observe is that most of these truncations affecting GLUN2A and GLUN2B, so or GRIN2A and GRIN2B genes, indeed, they are disease associated. While those affecting GRIN1, 2D, 3A, and 3B are mostly non-pathogenic, okay? So when, in other words, when there is one copy of GRIN2B or GRIN2A gene that is cut, this will lead to pathogenesis. We then characterized using these electrophysiology recordings or cellular experiments. And what we observe is that all these truncations, indeed, they correspond to a loss of function variants. And this is pretty important because nowadays, more than 100 different mutations, truncations have been described. And this means that with this study, we can already address more than 100 GRIN variants and provide this stratification for all of them. So once we have this functional annotation, as I mentioned before, the next step is to try to develop precision medicines. So I'm going to focus on a particular type of approaches that we have developed along the last years. And the rationale is that if we have an NMDA receptor that is somehow hypofunctional, meaning that it doesn't work that much as it should do, and what we want to do is to boost this NMDA receptor that is a loss of function, okay? It is working not enough. So we want to push them, to put some gas to the engine and to accelerate to somehow compensate this hyperfunctionality. And this is something that we achieved by means of using this serine in vitro and in individuals by providing L-serine into the diet. So by several experiments, we first, our pipeline is to initially start with what we call the preclinical studies. We go to the laboratory and we evaluate whether this therapeutic strategy is working in vitro, okay? So we first validate that this is working, and this is something that we published last year. And then with these results in hand, we contact the clinicians And we consider if we can translate these findings to the clinical practice. So with the proof of concept studies, currently we are together with the physicians from Sant Joan de Déu, Dr. Garcia Cazorla and Natalia Julià. We started a clinical trial with Herziening that will be presented by Natalia Julià. And we also are exploring 2 additional compounds called CAT23 for loss of function and ERK12 for gain of function. So we don't want to just fix on Herziening, but some other strategies are currently in process. It is important also to note that these compounds are all of them approved by the FDA or by the European Medicament Agency, meaning that they are very quickly translatable to the clinical practice. So I think that this was what I wanted to give you as the overview of NMDA receptors. I just want to fix some ideas and to tell you that we must understand NMDA receptors. Besides this stratification, I think that one of the next steps will consist in to understand how these brain phenotypes are connected with many other systems in human body. And I would like to end up by acknowledging all the people that has been rowing together to reach these goals and future goals. So especially people that we initiated these projects at IDIBELL, but now we recently moved to the University of Barcelona. So I already mentioned Ana Santos, but there is also Silvia Locurice, who's working with new experimental models of GRIN-related disorders, David Soto and Fede Miguel, Mireia Olivella and Adrián García for all the computational studies, and of course the invaluable help from Sant Joan de Déu, Àngels García Cazorla, Natalia Julià, and our collaborators in São Paulo and in CERN in France. And last, I would really also like to acknowledge the funding bodies, especially Precipita. This was an action that was performed last 2 years ago, and that allowed to provide the fundings for the annotations of up to 30 GRIN variants. Also the Spanish Green Patias Association, and also the individual donations that have been really important to achieve the GRIN-database and these studies on the truncations. And very importantly, I really, and on behalf of our team, we want to acknowledge all the people, all the families that are really supporting us, and you are the real driving force of all our research. Thank you very much, and I hope that next year we will have a physical meeting, and we will really share time together. Thank you very much.
Thank you very much, Xavi. That was a fantastic overview and presentation, as always. I don't want to jump in. We have already gotten a few questions. We will address them after Mireia finishes the first block. So I would like to just give over directly to Mireia. Thanks again, Xavi.
Thank you so much.
Okay, so are you able to see my screen?
Yes, we see it.
Okay. Okay, so thank you very much to the organizers. I have prepared some slides regarding GRIN-database, which is a new database that we have created containing genetic, clinical, structural, and functional information on GRIN variants. So this is our DNA. Our DNA is made of 3 billion nucleotides long, is made of 3 billion letters in a 4-letter alphabet, made of A, T, C, and G. And there are differences in our DNA. There are some letters that are different between us, not many at all, but this difference make a difference also in how we look alike to our predisposition to disease. And if we can have, for example, an adverse drug reaction. And this is very highly related to personalized medicine. And this is because we have been accumulating lots of mutations for thousands and thousands of years, and we are still accumulating mutations. So we are still changing our DNA, we are still changing the letters of our DNA. Every time that the baby is born, the baby will carry an average of 60 new mutations on the DNA. And these mutations do not come from the DNA of their parents. So we are generating changes in the DNA constantly in every baby that is born. So the DNA is the book that we need with all the instructions for any organism to live. And these instructions will be for our whole life. If we would like to store all this information on our DNA, we need all these books with the DNA. And inside these books, we'll find words and words. Well, it's just a single word because it's just with no breaks, with just a combination of 4 letters, A, T, C, and G, and this is called our genome. So we'll need all these books, like all this big bookshelf with all these books to store our DNA. And then behind an individual, you have to imagine that we have this bookshelf, and this bookshelf with these books, with a sequence, it's unique. If we compare the books from one individual to another, we'll realize that there are some differences in some pages. Not a lot of differences, but there are some difference between one individual to the other. And these differences can be in one letter. So it can only be one letter that is changed between one individual and the other. If these mutations, if this change in one letter falls in a position that is not really important to read the instructions of the life, so to read the DNA, then nothing will happen. The variant, the mutation will be just neutral. Or maybe it will be related to how we look like, but nothing will happen. If the variant, if the mutation falls in a region that it's kind of a little bit important to understand the instructions of life, then this may be related to a predisposition to disease. And then finally, if the variant is in a region where it's really, really important to understand the instruction, this can be disease causing. But there will be lots of differences between us and nothing will happen. Why a single change in the DNA can be disease causing? We have this piece of DNA here, and as you can see, a change in one letter of the DNA, just one letter, can be translated into a difference in the shape of the protein. So it can make a little difference in the conformation, in how the protein works. Maybe this change will cause that the protein won't be able to fold, to exist. And then if the protein is not able to have the proper shape or even fold completely, or just a slight change, this can make that the protein will lose how it works. It will have maybe a loss of function, or maybe the change in the shape of the protein will give like a gain of function. And then if this protein is important, is essential for the cell, then this will be disease causing. The last years, the cost of sequencing has dropped so fast. So like the first human genome, we were able to sequence it in 2000, and it was really, really expensive, and it took 11 years to sequence the first individual. But then like 10 years ago, the cost of the human genome was around 10,000 euros. And it took a lot of time, but now the cost of sequencing, of exome sequencing now, it's around 900 euros, and it takes just some hours to to get the sequence of any individual. So the cost of sequencing and the time of sequencing has dropped so fast, and this is making a big change. Thus, there are lots of sequencing projects around the world. We are in the era of what we call the big data. We want to understand every difference in our DNA and to compare different individuals around the world to understand the difference between us and how this can be related to disease and how can this be important, for example, for personalized medicine. And this represents a huge amount of data. This is why we need bioinformatics to store, to analyze, to interpret, to understand all this data. So as the cost of sequencing is dropping so fast, and it's not that expensive and time consuming as it was 10 years ago, now kids with neurological conditions with tentative clinical diagnosis are being sequenced, complementary to clinical and biochemical analysis. So then we get the sequence of the DNA, and sometimes we can see differences in their genome, in their DNA. And sometimes these differences in the DNA, in the genome, can be in a green gene. Still, these differences, these changes of one letter can be neutral, so nothing will happen. But sometimes these differences in the green gene can be disease causing, can be damaging, can make a change in the protein, and then can be related to pathology. So once a clinician, child neurologist identifies that there's a difference in of one letter of so many letters in a green gene, it's important to be really sure that this difference is the one that is related to the neurological condition. So to make sure that it's not a neutral mutation, a neutral variant. And then once we know, or once we are sure that this is damaging, is disease causing, then it will also be important to understand why is it disease causing. Is it causing a loss of function? Is it causing a gain of function? In order to go next, to move forward into the strategic therapy. So this last year, we have been contacted by more than 150 families. I think that we are around 180 families right now, indeed from families, but also from GRIN families associations, for worldwide child neurologists, and from child neurologists from the European Consortium. And the requests were about a variant, a mutation identified in a child with a neurological condition. And the requests were asking to classify these variants are really disease causing or not. And then if the variant was disease causing, they ask us if it was a loss of function or a gain of function. Before carrying the experiments in Xavier's lab to check if it was a gain of function or loss of function, we first need to explore if this mutation if this variant has been described before, because there's a lot of information regarding GRIN variants. So first we need to check all the available information. So some years ago, what happened was that when trying to explore all databases and all the information in these GRIN databases and some non-specific GRIN databases in terms of pathogenesis and functional annotation, we realized that all the information were split in several databases and also in the scientific literature. So it's like having all these books with all the genetic information and also experimental information split around the world in different files in different countries. And in addition, when comparing the information from different databases, we realized that the information was not exactly the same. So we have some variants that were classified in a different way depending on the database. And information was growing so fast, and there were variants and more variants each day. So at the end, it was not easy to check for all the information before carrying the experiments. So this is why we decided to construct a new database. The aim was to construct a unified and curated with all the information integrated GRIN database containing all GRIN variants. We spent like weeks, I think that month, curating the information entry by entry, extracting all the information from the scientific literature. And well, this database was created by Adrián García. He's a PhD student. And it was possible because of the collaboration of all GRIN families and the financial support that we received from the Spanish GRIN Association. And we have been using this database for more than 1 year in order to give answer to the request from different child neurologists. The idea is that we have in just some seconds to be able to display all information that we have on a variant. So we can give a quick answer to the child neurologist regarding the pathogenesis and also to the functional notation of a variant. Right now, we have a total number of variants, which is 4,473 variants on GRIN genes. Most of these variants are on GRIN1, GRIN2A, and GRIN2B genes. But from these 4,473 variants, we only have 444 variants that are disease-causing, that are damaging. But there are different, looking at these pathogenic variants, these disease-causing variants, we have 3 different scenarios. For some of these variants, the variant is fully annotated, so we can give an answer like, yes, it's a loss of function or it's a gain of function just in seconds, just in the time that takes us to answer an email. We can tell the clinicians that the variant is partially annotated, so further experiments are required, and sometimes the variant is not annotated. So let me show you how the database works. Sorry that I will move this down. Okay, so we just introduce something that it can be easily found in any genetic report. So we have the name of the gene, then the position, and then the amino acid that is mutated. So we introduce just the name of the gene, the position, and the rest is something like this that can be easily found in any genetic report. And then in seconds, we just obtain this answer, which is all the information integrated from different databases. For example, in here we have that this variant has been described in CSVER, in GRIN-Leipzig, in Barcelona GRIN, and in LOB-D. So this variant has been described in these 4 databases. And then we have information regarding on all the experiments and where these experiments were, these experiments were performed in 2 different labs, in CFER and in Barcelona GRIN team. Then we have a clinical descriptions, also integrating information, in this case from CFER, from GRIN-Leipzig, and from Barcelona GRIN. And at the end of integrating all the information, especially the experimental information, we can conclude that yes, this mutation is, this variant is disease causing, is disease associated, and this variant is a loss of function. But sometimes we just introduce a mutation and then we realize that it's predicted as a gain of function, as the functional information is not conclusive. Then I just call Xavier and ask please to do some complementary experiment just to be sure that this variant is really a gain of function. And sometimes we introduce a mutation and we realize that there's no information, that this variant has been described in patients with a neurological condition, but there's no functional information. And then we have to start from the beginning, trying to annotate this variant. This can take some weeks. And sometimes it happens that we receive the variant, the GRIN2B, the position, and the change. But when we run the database, then we realize that this variant is found in population with a high allele frequency, and that this variant is neutral, is not related to disease. And this is really important to tell the child neurologist that this variant is not the disease causing, and then that the neurological condition might be related to another gene. And then it's important to keep on looking for the cause, for the diagnosis. Okay, so now let me move to the structure, as I am a structural bioinformatics, and well, I look a lot to the structure. I think that they are really important to look at to understand what's happening. This is the NMDA receptor, and this is the extracellular region. This is the ligand binding domain. I'm sorry, and down here we have the transmembrane domain. So when we receive a variant, we also look to the structure of the protein to try to understand what's going on, why this variant is making this change in the shape of the protein that at the end will be related to the neurological condition. So when we receive the request, we always make a molecular modeling, trying to understand the position, the changes of the residues, and how these changes are making a new shape, are making new interactions inside of the protein, and then we integrate these with the experimental results from our lab or from other labs, and to the corresponding clinical phenotype, if it's mild, if it's severe, and well, to different things, to give a unique and integrated view from the structural to the genetics to the functional annotations. No, everything should be integrated, and we have a view of what's going on. Every time that we receive a request, we collect the information regarding the clinical phenotype of the child and the genetic information, and we enrich our database with the results from the experiments in Xavier's lab. And by integrating this information of more than 4,000 variants, we have been able to kind of understand a little bit more the effect of this variant on green genes. Here we have a plot. In green are neutral variants on green genes. As you can see, there are more neutral variants than pathogenic or disease-causing or disease-associated variants. And in red, we have these disease-associated variants. We can see that there are some regions in the NMDA receptor, in the green genes, that if we change, nothing happens. So if we just change the letters of the DNA, that will result in a change in the amino acids in this region, nothing will happen. And also nothing will happen usually in the intracellular region. But if we just touch a little bit the regions around the ligand binding domain or the transmembrane region, these regions are sensitive. So it's the regions in these books with the DNA that are essential to read the instructions to live. And then these regions are really highly sensitive, are really essential. And then if there are some changes here, then the disease causing. Also, we have been able to map to classify the variants as loss of function and gain of function, and to map them on the sequence and to map them on the structure, and realizing that there are some regions that are more related to gain of function and some regions that are more related to loss of function. And well, helps us a lot to understand what's going on and to make sense when we look at the results of the experiments. Now, why is it a loss of function or a gain of function will be highly dependent on the region on the NMDA receptor. And also, and this is important too, we have been able, by collecting information from the clinical phenotypes of the patients and of all the information in the scientific literature and in different databases, we have been able to see some differences in the clinical phenotype between GRIN-1, GRIN-2A, and GRIN-2B. So there are important differences, and also there are important differences, especially in GRIN-2B, for example, and also some differences in GRIN-2A and in GRIN-1 for loss of function or gain of function. So the clinical phenotype seems that there might be important differences that will be subunit dependent, so gene dependent, and also gain of function or loss of function. And this is important because this can help clinicians to characterize these green childs, and then to see if the clinical symptoms are in according to the different classifications and the different clinical phenotypes related to the green subunits and also to gain of function or loss of function. But right now, this information regarding the clinical phenotypes has a lot of limitations because this information has been collected for lots of years, for this for 10 years maybe, but under different criteria. Some are come from child neurologists, some not, some from geneticists. So there's a lot, this information have some limitations. I think that it's really important, and it's urgent now, that we have a patient registry that is collecting the information regarding the clinical phenotype of GRIN kids. But under the same criteria, this will help us a lot to understand the differences in the clinical phenotypes, and this can help then once integrate and analyze and after interpreting this data to help clinicians with the diagnosis of GRIN kids. Still, we have 150 GRIN variants for which we don't have any functional annotation. So for these variants, we need to carry experiments from the beginning. And these 150 variants represents hundreds of patients. We don't have a database of patients. We have a database of variants. So we have to understand that behind each variant, there might be more than 10 patients or 20 patients. So still we have these 150 variants for which we don't know the functional annotation. And it's important because this is the first step to design a strategic therapy. And behind these 150 variants, there will be hundreds of patients with this variant. But this is expensive and it takes a lot of time. But I think that we have to put a big effort in annotating these variants. Okay, so this is the GRIN database. The aim of this GRIN database is just to give a tool, not to us, to the Barcelona GRIN team, but also to clinicians and child neurologists around the world to quickly and easily check if the variant is a gain of function, loss of function, and if it's disease causing. And also for families to check if the information regarding the variant is in the database or not. So Barcelona, this project has been done by the Barcelona GRIN team. We have the clinicians, the child neurologists in Sant Joan de Déu, Àngels García Cazorla and Natalia Julià. Here we have in the University of Barcelona with a neurobiologist and the electrophysiologist, Xavier Altafaj, David Soto, Ana Santos, and Federico Miguel. And in the University of Vic, we have Universitat Pompeu Fabra, the bioinformaticians, Adrián García, and myself. And I would like to thank you very much to all our collaborators and partners that have made this project possible. Okay, so thank you very much. And Christian, I don't know if now we have time for questions.
Yes, thanks, Mireia. Thank you very much for your presentation. I think we have a few questions on the question and answer board, but I'm sorry, we were having a little bit of trouble. So if somebody has any questions that for whatever reason cannot ask them at the Q&A chat, please put it in the regular chat so we can look at them. I'm going to start with maybe the easiest question of all, then the other three that are in the chat, they're a little bit more complicated, but I would like to start with a question from one of our attendees who they actually just wanted to know how that we come about doing the functional testing, if you need blood, and the other question together with this was if anybody is undergoing any kind of treatment and what are the results.
Yes, regarding the functional annotations, we don't need blood samples or any kind of samples from children. We just need the genetic report. So normally when the genetics unit or department will give you the result from this genetic report with a code number of the gene that is affected and these milestones. So normally there is the position, the nucleotide position, and the amino acid that is affected. So this is the only thing we need to check in Mireia's database if the variant has been already described or not, or if functional studies need to be performed. Nevertheless, there is this informed signed consent that is necessary, because since we will have your confidential data, we need your approval that you agree and signature that you are agreeing that this information will be kept confidential, of course, anonymous, but we are able to process this information and use it for research goals, okay? So only send this genetic report and the signed consent form. And regarding the treatments, I think that this will be the object of Àngels García Cazorla and Natalia Julià. So I don't want to spoil the talks, but there are, I think that something very important, and I want to state, is that there are already the first clinical trial that has been initiated in Barcelona's Children's Hospital. And this is a real milestone in the field of GRIN-related disorders. So this is something really amazing that they are performing. And this is a very important piece of hope for GRIN children. But I think that Àngels can give you a very, a lot of details on that.
Okay, so I think there's another question regarding the database, which is how do you make sure that people are not registered in more than one place and therefore possibly being double count, yes. This is the database, our database at least, is not a list, or it's not a database of patients. It's a database of variants. So behind each variant, there can be several patients. So it doesn't matter. We usually find that the same patient has been registered in different databases. And this is no problem for us because we are just collecting variants. So just the list of variants. It doesn't matter if it's from one patient or from 10 patients. And then we just describe if it's pathogenic or not, and it's a loss of function or a gain of function. So don't be afraid to give the information to several databases. But I think that something different is regarding a patient registry. And a patient registry is really important that it's a list of patients, and then they will have an identifier, and you should make sure that there's no double entry for the same patient. But it makes a big difference, a patient registry to a database of GRIN variants.
Thank you. There's also 3 other questions that were at the beginning of the list of the Q&A. One of them is why the NMDA dysfunction is necessarily leading to loss of function versus or gain of function. Couldn't it be a combination of both?
Yes. Yes. When we try to understand biology and what a mutation generates, we are humans, and we have this boxed vision of reality or of biology. So in our mind or our brains, we try to make like different classifications. So we roughly classify into loss or gain of function. These are the main categories, but this is like a spectrum and a gradient. So not all the mutations will be 100% loss of function or 100% gain of function. There is a between, a penumbra area where you have some alterations that correspond to gain of function, but the same variant has some other aspects that are from a loss of function. And this is like a third category that we call it the complex variants, which are pretty difficult to also design therapeutic strategies, because they are not that pure as a law of 100% loss or 100% gain of function. But yes, totally agree that there is this myriad of GRIN variants. But for the sake of getting categories, we classify in these 2 main categories, loss or gain of function.
I'm just going to continue with the questions. The next one is regarding puberty, and if puberty changes what we're seeing in terms of loss of function, because it's been commented some families that that the kids seem to improve as they reach puberty, and maybe this goes more with a change maybe of the proteins and the connectivity of the brain in terms of the number of, for example, this is the case of a GRIN2B child. You know, could it be that the GRIN2B changes into a GRIN2A? And I don't know if you can comment something about that.
Yes, that's an important feedback from families. That is something that we didn't explore yet. Regarding this developmental shift that has been described, I think that Angels has, she has been preparing a description of this, but at least the different GRIN genes are expressed at different stages of development. So at the beginning, GRIN1 and 2B are widely expressed, and then GRIN2A starts to expression. As far as I know, during puberty, there is not an additional shift, but probably hormonal control and crosstalk with the brain can have an effect. And well, I think it's better if a physician and a child neurologist can give you a more appropriate answer. But yes, it makes sense.
Yeah, and I think the next question also might be more related to for Angels, but because again, this is about experience of having some of these kids either have low or high sensitivity to anesthetics, and they could be explained by the NMDA-receptor. I don't know if you want to comment or you want to leave that for Angels.
I think we can leave it for Angels, yes. So some kind of anesthetics, they work on also the ketamine-based anesthetics. They have an effect directly on NMDA receptors, but I don't know about green children that have been anesthetized. What are the responses if they are less prone to or more resistant to anesthetics, or which kind of anesthetics?
Okay, so I'll go for the next one. Is there a difference regarding research and possible clinical approaches between missense and nonsense variants?
Mireia or myself?
I mean, like Xavier was explaining, I think in his presentation, making nonsense proteins usually gives a loss of function protein, while with missense variants, we have gain of function or loss of function, or at least with all variants that we have seen that are nonsense, they at the end result in a loss of function.
Yeah, so what is really clear is that these pathogenic GRIN nonsense mutations, they result in a loss of function. And what is interesting, and it's something that I skipped from the presentation because of time restrictions, but the phenotypes are milder normally. So this is like a general rule, but in general, the phenotypes of GRIN children with GRIN nonsense or truncations are milder than those that have missense mutations. So this is somehow like having less receptors, because at the end, nonsense mutations trigger a reduction on NMDA receptors. So these children have less receptors, but at least all the receptors they have, they work properly. So it's a matter of number. There is not enough NMDA receptors. What is happening in the context of missense mutations is that in most of the cases, these mutations, they form already NMDA receptors, but they are misfunctioning. So they are interfering with, let's say, normal NMDA receptors, and they have a very powerful effect even in the normal or wild type receptors. So it's like a more severe. This is like a general rule. Yes.
Thank you. And the last one of the questions that we have right now, if anybody wants to put something else on the chat, we might have a couple minutes, is there a web link for the database that Mireia just showed?
Yes, there's a web link. We are just publishing the paper and waiting for some answers from the editor. I hope it will be in 1 week. It has took us a long time because the database was finished in March, and now the process of publishing is being so slow. But once it's published, then it's published, it will be in maximum 2 weeks. We, of course, with the link to the GRIN community, so it can be used and for families, from child neurologists, from anyone. This is the idea, to share it, and that this information can be used from anyone. So families will be able also to check if the information is in the database. As this database is collecting information from all databases, then just by getting inside the database and introducing the protein and the chain, they will realize if it's really in any of a database. And if not, and if they want to give the information regarding the variant, they can contact Barcelona GRIN team. I think that there's the address somewhere they can contact in order to request for a variant that is not there. And still, while we are waiting from this link, they can also request and write an email to us to request for any information regarding any variant.